Okay, welcome everybody for coming, or thank you for coming to Disruptive Thinkers. Hi, Emilio. Thanks for coming out tonight. I see a lot of new faces, I think. Who's first time here tonight? Okay, awesome. Thank you for coming out. We have a great talk tonight. We have Dr. Eric David from Organovo who's going to explain to us what Organovo does, and Paula Mellon's going to talk to us about what she does as well. Start out with. First, let me tell you a quick couple of things about Disruptive Thinkers, how we started, what we aim to do, and that sort of stuff. Disruptive Thinkers was founded by Lieutenant Ben Coleman about a year and a half, 2 years ago, and just kind of he and some friends in a bull session talking about how the Navy needs more disruptive thinkers, right? And for those of us who have dealt with government or military or even any large organization, there tends to be that stagnation and that bureaucracy that sets in and makes things difficult to do. So what we do is we try to bring together a diverse group of people that want to learn different ways to do things or to search best practices, and that's what Disruptive Thinkers is. We've done some awesome talks so far. Tonight, 3D bioprinting, very disruptive technology once that gets going. And you can go to the hospital and get a new heart printed for you. Just a couple things about what we've been doing and what Lieutenant Coleman has been doing. We've been joining we've started a Navy thing called the CNO's Rapid Innovation Cell. So that's Disruptive Thinkers has got a military arm now as well. And that's awesome. We actually just traveled to DC last week, got to meet with the CNO and some other very interesting people. And so the CNO, for those of you who are wondering what the military jargon is, it's Chief of Naval Operations. He's the top admiral in the Navy. And so those of you who are friends with Ben, you know it's got to be pretty exciting for him to be in that spotlight now. And that's where he's going with this. So Disruptive Thinkers doing some awesome things. Okay, we're going to start off with a short talk by Paula Mellon, and Paula is going to talk, just to give you a setup of what we're doing here, we're going to try to do a little 5-minute blast from various disruptive thinkers every month. That way you can get to know one another better, find out what strengths another person might have, and maybe you find there's a business in common you have, or something that you, I've always wanted to meet somebody who works in this industry, and there you go. So this is to help us meet people more easily. And Paula is a ex-South African entrepreneur, and she's started and owned several businesses, and she moved to USA. I don't know, how long ago did you move?
A year ago.
A year ago, and to continue her entrepreneurship. So.
Hi everyone. As Jeff kindly introduced me, Paula Mellon, ex-South African entrepreneur. Started and owned several different businesses in South Africa. Started my career at the age of 23 in the realty industry. It was, what I started was a kind of an online search network realty business where everybody could put their information onto a database, and all the realtors and the residential homeowners, commercial owners, industrial owners all grouped together and were able to search and identify property that was most suitable for them. After that, I worked at an engineering company and was in the management executive team. What happened was the owners of the engineering company immigrated to Australia, and I landed up doing a management buyout. It was a very male-dominated industry, and it was pretty cool because they had very innovative products that they were making for the automotive industry, electrical, all kinds of verticals in South Africa. And what happened was we decided to focus on our own product, and I patented a hybrid electrical cable gland, and also improved the shroud that is a protective covering for the electrical cable gland, and took that product to market. That allowed me then to exit the engineering industry, because I've been an entrepreneur. You'll probably, if any of you are, you'll find out that you kind of get bored with certain things and you like to move on. And that's what happened, is I got a little bit bored with engineering, exited engineering and went into the information technology industry, which brought me actually to San Diego. So back in 2006, I arrived here, went to the Desktop Linux Summit, identified a very niche product there, that I negotiated exclusivity for the whole of Africa and took it into 54 countries in Africa. So started an import company and a distribution company. Did very well for the last 6 years. Unfortunately, crime is getting very bad and rampant in South Africa, and had another terrible death kind of home invasion situation. And that's kind of what solidified my decision to leave the country and come back to San Diego, where I'd fallen in love with back in 2006. And luckily, I still had my contacts from back in those days. And yeah, the last year, I've been kind of got a whole client base together. So I do business servicing and consulting for various different companies that are either in the fitness industry or technology industry or biotech industry, online businesses. So I've probably got about 4 clients at the moment that I'm working with, and helping them with their back end, getting their product to market, or resourcing a team that they need, or researching products that they don't have the time to do. So I go in and do kind of like the headache work for all of them. And yeah, that's who I am. So, you know, when I joined Disruptive Thinkers, because I resonate with the philosophy of the team, I said to Jeff when we came to the TED Talk, well, you know, we come here every kind of once a month, and sometimes we get to meet the 1 or 2 people, and we go away and we go, okay, so-and-so does this and so-and-so does that. But it would be great to actually know what everybody does, that if I've got a project going and I know Jeff specializes in this, I could go to him and kind of recruit a team or get some information from Jeff and collaborate and get kind of the whole project moving along. So that's the whole idea about every now and then having somebody of Disruptive Thinkers come up and give a talk about who they are. Thank you.
Outstanding. Well, thanks for being the first one to tackle that too. We were gonna have one of our pilots talk about his Osprey adventures, but unfortunately he is flying tonight, so wasn't able to talk about it. Aaron Odman. Yes. No, I just dimed him out in front of the whole world. No, he'll be here next time. So, just real quick introduction to Dr. Eric David. He's the Chief Strategy Officer for Organovo, and I've noticed that he's both a doctor and a lawyer, and that's pretty amazing. So, and from what I'm talking to Eric, he's been with Organovo from the start, and he's going to talk to us today about what they do with 3D bioprinting and all the excitement that's going on there. So, let's give him a warm welcome.
Well, thank you guys for inviting me. I'm very happy to be here. I'll just sort of tell the story in free form of what the company is and does and how we came up with the idea of what the motivation was. In terms of disruptive thinking, you'll see from my background as a doctor and a lawyer, I sort of believe in bringing very disparate fields together. I think you get a much better solution in a venue like this, where you bring people from completely different backgrounds, different sectors together to solve problems. So as you gather from my background, I sort of, doctor and a lawyer. Why did that happen? Well, fundamentally, I couldn't decide what I wanted to do. I had a deep interest in sort of business and law on the one hand. I had a deep interest in science and medicine on the other hand. I grew up in a family full of doctors. Everyone told me, whatever you do, don't go into medicine. You won't be able to practice medicine the way you want to practice. It's changing. Don't do it. So I listened to them, and I went into law for a little while, and I realized I wasn't entirely satisfied. And I realized I kind of wanted to be at the intersection of these two, of business and medicine, law and medicine. So I worked at, I became a doctor, worked in a hospital for a while, did a lot of research in immunology and cancer, infectious disease, and I started, you come across a lot of problems as you do research and practice in a hospital. And Lord knows, you pick up the paper on any given day, and you're confronted by 1,000 different problems in the healthcare industry. But there were a couple that sort of stood out to me that I became very interested in. One, first of all, was, I'm someone who, as I said, I grew up in a medical family. I firmly believe that there are pharmaceutical and biopharmaceutical and really innovative solutions. I believe that it's an industry with a great history, sometimes a problematic history, but what industry doesn't have a problematic history? But R&D is really tough, and it's only gotten more expensive. In fact, if you sort of map R&D spending over the last 20 years or so, you'll see R&D spending just keeps going up. And new drug approvals kind of went up to a certain point in the mid-90s. They can be a little bit cyclical, but they've kind of tapered and even gone down. Although I will say 2012 was somewhat of a banner year in the last decade. But all in all, spending on R&D has far outpaced new drug approvals. So we're spending more and more on R&D, and we're getting little innovation. And why is that? Well, a lot of different reasons that we don't need to go into here, but one very important reason is drugs often fail in late-stage clinical trials. We're talking, by the time they've been in patients for a while. You don't know enough about them in the preclinical when you're just doing, tissue culture work, which is basically cells in a petri dish, or animal models. So then you put them in patients first in low dose, largely as a safety trial, and then in larger scale trials. And drugs kept failing and keep failing in late phase 2, early phase 3. By the time to give you an idea of what that means, by the end of preclinical, you've probably spent somewhere between 200 million and 400 or 500 million on a drug. So if you fail in preclinical before you ever get into human beings, you spent a whole lot of money, but not as much as you're about to spend. Because from the end of preclinical to mid-phase 2, early phase 3, you've probably spent about 800 to $1 billion just on that part alone. So obviously you want to fail as early as possible. And that's easier said than done, right? Because it requires you have animal models that are more predictive, or tissue culture models on human cells in a petri dish that are more predictive. So that was one problem that I kept looking at. The other problem that you go from sort of the lab to the clinic, and obviously, as you all know, organs for transplantation are a pretty scarce commodity in any nation, and so there are lots of problems to solve around. How do we get people solutions that are either, if not whole organs, then enough functionality in an organ to do the job. So those are two problems that had been on my mind for a while, and I was living in New York, and I was about to start work for a consulting firm, and about a week or so before I think I formally started, I was there and met, there was someone else who was starting who came from a finance background. I just finished my medical residency. I was coming from that background. Someone who had been working at Citibank was talking to him. His name was Andros Forgax, and he found out I was a doctor and that I was coming to consulting from there. And he said, listen, my dad is a biophysicist, and he does some really cool work. He has like this crazy printer that can print cells and tissues. And I just, he gets approached by venture capitalists and investors all the time. I just wondered if you could talk to him and give him some advice on how to commercialize it. And I said, yeah, I'd be happy to. So I met him for breakfast a few weeks later, and heard about his technology, and it was quite remarkable. And went home and sort of read a bunch of his papers and did some diligence. And I kind of said, well, if this does what I think it does, then maybe I want to help you commercialize this. Maybe I want to play a role in it. So I started thinking about the best way to roll this out strategically, and we'd have these Sunday night phone calls with Gabor Forgax, who's the scientific founder of the company, Andros, his son, and then I asked a friend of mine, Keith Murphy, who'd been at Amgen for about a decade, said, I don't know, I've got this cool technology, maybe you want to join these calls and we'll see what comes of it. So we'd all get on the phone on Sunday nights, and we started to develop a strategy, and Keith eventually left Amgen and became effectively the first employee of the company, and he drove it forward for a long time, and I came on board. Even though I was one of the co-founders, I didn't come on board full-time until just about a year ago. And I can tell you a little bit more later about the financing, and for those of you interested in how you finance a company like this and get it from just a Sunday night phone calls to an actual company. But let me tell you a little bit. I've told you about the need. Let me tell you about how this technology solves the need. So 3D bioprinter sounds great, and most of us are familiar with printers, and so you think, okay, wow, a printer that can print human organ tissue. That's great. Print me a liver, print me a lung, print me anything. And that obviously is that's the ultimate vision of the company. I can't print you a whole organ right now, but I can do an awful lot. I can print parts of organs and functional tissue from different organs. But where does it all come from? How does it happen? Because you can't just take an HP printer and put it in a test tube and expect that it's going to print you organ tissue. So what Dr. Forgax's critical realization was, he didn't set out. No one really sets out with the idea of like, I'm going to invent a printer that prints organ tissue, right? But he was studying mammalian development. He was studying how cells come together to form mammals like us. And he observed something really interesting. He said, it's not that our bodies are not built one cell at a time. It's not one cell, then two, then three. After a certain point of development, it's that there are droplets of cells, droplets of a few thousand cells here and a few thousand cells there. Those droplets are sort of come together with different cells, and they coalesce, and the cells then do the rest of the work. The cells get the right cues to self-assemble into these very fine microscopic structures. And that's how our bodies are built. And so he thought, gosh, I wonder if I can help that process along. I wonder if I had a printer that printed out droplets of cells, would this effectively replicate the process of organ development? And that's what he began working on, and that's what the printer does. The ink, if you will, in the printer is not individual cells that are laid down one at a time. The ink is composed of these droplets of cells that coalesce, and we rely on the cells to then do the rest of the work from there and build these tiny structures that are on such a scale that a printer couldn't really get there. And so that's what we do. And the printer, we print tissue, as you can imagine, to solve the two problems that I described earlier. So one is better models for drug discovery, right? So if you go to a pharmaceutical company doing R&D and you say, okay, I can print you tissue from any organ, any disease model that you want to work on, just let me know, and I can print you some tiny pieces, and you can do drug testing on it. That's to work with that company to validate the fact that this tissue does in fact behave like real human tissue. But once you do, then you have an extremely powerful model. And so that's one of the things that we do right now. And the other thing is, of course, gosh, how can we develop organs for transplantation and implantation that are tissues that are themselves therapeutic? And as I said, an organ is, as you can imagine, so just inherently complicated that right now I can't print a liver that looks just like my liver and give it to a transplant team to put into a patient. But what I can do is start to print smaller pieces of tissue of liver that have many of the cell types that you need to do all the work of the liver, and they're in an architecturally correct context. So if you look at it under a microscope, it looks very much like the liver tissue in my body, and and I can make enough of that to replace some of the function. And the truth about often people who are sick enough to need organs, you think of people who get kidney transplants or liver transplants. You're not taking that patient from 0% function back to 100% function, right? You're taking someone who usually has fallen somewhere below 15 to 20% of their function, right? That's when patients with, with diseased kidneys and diseased livers really get into trouble. You're dropping down to like, 10% of their normal function. That's where they really need a transplant. And the transplant will often get them to, if you're lucky, 25% of their original function, or 30%. But that's enough that they can live, a long time on that. It's enough to give them their lives back. And so that's what we hope to be able to do with many of these tissues. We haven't yet implanted any of our tissues into people. That's that's something we're working towards. It's Organovo is now a company of about 30 people. It is mostly R&D folks, and and we work all the time to the real art of it now is thinking, okay, well, how do we first of all, there's art on the business side. What's the strategy? Which we don't have the bandwidth or the money to pursue every organ in the body. So how do you decide which organs you're going to pursue, both for a sort of drug testing standpoint, but also for a therapeutic standpoint? How do you read the market and look ahead and map not only what you think your technology is going to be best suited for, but what the market's really going to want? Where is the real unmet need? Where is the real demand? Where there are currently no good therapeutic options or no good disease models? And then there's a lot of art on the science side, right? And this is, it's one of the things that I think everyone in the sciences loves about science is I think the way science is taught, at least in the U.S., I can't speak to other countries, but in the U.S., science and engineering are sort of locked together, and inherently they're looked at as these uncreative things that are largely technical that you can fundamentally just learn from textbooks, and that's unfair, right? No one would spend their life pursuing these things if they were just, if it was just sort of, arithmetic, right? It's an intensely creative field, and so there's a great deal of art to what our teams do on the R&D side, and I make it sound very simple by saying you have a printer that puts these droplets in the right place and lays them down correct, in the correct architecture, and then the cells do the rest of the work. But backtrack from there. Well, how do you give the cells the right cues? What cues do they need? What are the right combinations of cell types that you need to put in? How do you tell the printer where to put the cells? How do you prepare the ink that we call bioink? All of the, when you replicate the liver, the liver has, I'll approximate, but it's probably got about 45 different types of cells in it. Well, if I'm going to replicate a functional piece of liver, do I really need all 45 of those cell types? Or do I just need, 5, 6, 10 of them? Answering those questions is really hard. These are questions no one has really answered before. Or people have answered them in different bits and pieces for different reasons, but not very often for the reason of trying to reconstruct functional organ tissue. And so there's a great deal of art on the R&D side, and that's, I think for me at least, that's the great fun of what we do, is when you are creating a new part of an industry, it requires a great deal of skill and creativity on the business side, on the commercial side, on the manufacturing side, and of course on the R&D side. And that's just, it's just inherently fun. I mean, there are all these problems that you solve, you seek to bring together people with very different backgrounds. Right? There are people who work at our company who have hardcore engineering backgrounds or bioengineering backgrounds or cell biology, molecular biology. There are people with much more business backgrounds who so it's you bring together these teams that are disruptive simply because they come from such different backgrounds. They bring very different things to the table, and I firmly believe you get a much better solution to a problem that way. So let me pause there for a second. I have no idea how long I was talking for. But it's probably a good place to stop and just say, first of all, Jeff, any part of the story that you want me to cover in particular?
No, I have questions for you, but
Okay.
I'll open the floor to
Yeah.
questions first.
Sure.
So we talked about this earlier. I'm actually also working at the intersection of knowledge and healthcare. My background is not biomass, so but then I figure, when you input something, you generate an output, right? There must be a model, there must be an algorithm associated with your printer, so first off, like you cannot inject, liver tissue and hoping you're gonna print out a kidney tissue, right?
Right.
So and then secondly, you gotta model the organ tissue that you wanna print out according to that, bioenvironment or the architecture of that organ or whatever like key cell types you just mentioned.
Yeah.
Earlier, right. So I think there's like two pieces of elements that are very critical in terms of that intelligence of it. One is like how do you model it, right? You have a model that's your baseline, and then you apply the algorithm, the intelligence to print it out.
Right.
Because it's not like mechanic printing like
Absolutely.
all the other 3D printers, just layers on
Yep.
layers and layers, right? Kind of like have to cultivate a culture. You have to
Mhm.
provide whatever an viable environment has to provide for this cells to kind of like, how do you say, replicate.
Yeah.
Right. So in terms of that, like I think your model must be tissue specific.
Absolutely.
It cannot be like just like all agnostic.
Yep.
Yeah. Right.
Yeah.
And secondly, your algorithm must be specific as well. Because like if I want to generate liver specific tissues versus kidney or heart specific tissues, those requirements, those environments are different.
Yep, absolutely.
Am I getting there?
Yeah, no, absolutely. You're right. And the software algorithms that the printer runs on are tissue specific. They're sometimes specific to the cells that you're working with at that time. And so yeah, it's and that again, there's a lot of art there, right? There's a lot of trial and error over just the last, since we founded the company in 2007, and even before that, there's a great deal of trial and error, and then you pick up hopefully in the trial and error for 4 or 5 different tissues. There are, as you might imagine, there are themes that are common across tissues.
Across organs.
Right, across organs and across tissues, and then there are aspects that are very tissue specific.
That's how you can optimize yourself.
Yeah.
The model.
Yeah.
So I read the news that you had a collaboration with Autodesk, right?
Yeah.
Are they going to be the ones that generate those models for you? What are the people that are working on that project, and what kind of backgrounds are they coming from?
So we're thrilled to be working with Autodesk, and yeah, they've been helping us design the interface for the printer. Do you guys all know who Autodesk is? Okay. So did someone say no? Okay.
Everybody else does.
Okay. No. So I mean, there's no reason you necessarily should. They're I mean, they are. If you've ever seen AutoCAD software, 3D design software, they're the company that I think invented it, and they're certainly, as far as I understand, I'm not a high tech person, but I think they're pretty much the leading company in the world with this, and it's remarkable, right? They do these incredible things where you design, it's that interface between 2D and 3D and how you translate 3D design into objects. Brilliant stuff that they do. And so, so yeah, we have a collaboration with them to help us create the interface for the printers. Up until our collaboration with them, it was largely, our internal engineers who, on their own or occasionally consulting with folks who knew a little bit more about software, had written all the scripts, and but again, it's all just people relying on their own ingenuity. But now to have a partner like Autodesk just makes it all the more satisfying. And it's a combination of people with software backgrounds, people with hardware engineering backgrounds, people with bioengineering backgrounds, all of that together.
Yeah, just to follow up that, I mean, it's very interesting because, in the software engineer, hardware engineer, you have somebody that's basically providing development kit
Mhm.
for most of your basic hardware products, like FPGAs, which is kind of what I think was
Yeah.
bio. You have to set design rules, and you have to have support. I don't know, other than PCR guys in biotech, nobody's really done that. So
Yeah.
I was going to ask you how you dealt with that, but it seems like you're working with Autodesk.
Yeah.
They have sort of, they're going to help you set up design rules. They know how
Right.
people design this stuff.
Right.
It's really-
Yeah, I know it's many years until you're actually going to be able to print the liver, but it's very intriguing in that many of the issues that you're dealing with. One issue is vascularization.
Yep.
How can you build blood vessels into into the structure?
Mhm.
And is it possible that the liver you finally come up with, it's not going to be liver shaped. It's going
Right.
to be a brick or a
Right.
cube that makes it so easy to get the blood in and
That's
out.
right.
It's going to be better than a liver. Is that, and are you already trying to figure out how are we going to, put a little valve on there to hook it up
Right,
to,
right.
to the rest of the?
No, it's a great point. I mean, I certainly would not personally be as audacious as to say it would be better than a liver, just because the more I think you study the body and see it in action, it's like, okay, well, everything was designed that, everything emerged that way, evolved that way for a reason. But I think you're absolutely right. There are several different schools of thought, and we're pursuing different avenues, right? One school of thought says, okay, if you can study the way the liver, at some point the liver was a much smaller number of cells that was given the right cues to develop into a full human liver. So should I try to start by putting together that smaller number of cells and let them just grow and flourish given the right signals? There's another school of thought that says, well, what if I start with matured cells and put them together into a structure that maybe looks not terribly like an actual liver, but as long as the microstructures are correct, I can create some different central structures that still get the, if you will, the inputs and the outputs right, but simplifies the gross architecture enough that it's easier to make and it can be scaled up and manufactured more readily, right? Those are the sorts. It's exactly the right question. Is you want to know, okay, not only it does the world very little good if we can make a liver, but it takes us 10 years to make it, right? So you want to be able to scale these up. You want to make it accessible to enough people. It's not only, frankly, the time to manufacture. It's, if you think, just the cost of cells and the access to cells. I mean, cells come from somewhere, whether they're induced pluripotent stem cells or whether they're mature liver cells. They've come from some human source, usually, in one way or another. And so it's also a scarce resource. It's not as scarce as a whole organ, say. So you need to be very careful from a resource perspective and a cost perspective as well.
Speaking of cells, where would you get the raw material? Like, let's say you were going to print something something from me to test something before you give it to me. What would you take? Take something from my body, like a skin sample, or is there a certain type of cell?
So again, it's another great question, and this is a problem that the industry of sort of tissue engineering, regenerative medicine faces as well. So you can approach it exactly those two ways, right? You can either take autologous cells from our own bodies, and put them back into our same bodies, or you can take what people call allogeneic cells, cells from somebody else's body, another source, and put them in. Either is workable, right? The overwhelming benefit, as you all know, to taking autologous cells is that when I put that new structure back into the body, I don't need to give immune suppression. The body recognizes it as my own. The downside is now I have to set up a whole infrastructure for harvesting those cells, taking them out, growing them up to the right cell types that I need, and then making the structure and then putting it back in, right? That's a lot of different steps. Allogeneic is easy because I can get the cells from a number of different, there are companies around the world that specialize in providing cells, and they get them from a huge variety of sources that we can go into offline. But they're not my own cells. The benefit is they're much easier to source and cheaper to source that way. The downside, among others, is, okay, if I'm going to put that structure now into my body, I've got to give some immune suppression, the same way I do when I put in an organ that was transplanted. And that creates, as you know, its own problems. So that's another big problem that the industry is grappling with. And for in vitro tissues, for the tissues we make for drug discovery, thankfully, that's not a problem we have to worry about. We can take cells from any source, and sometimes we want cells from patients with a certain very rare disease, and so we can get those cells and make in vitro tissues to do drug discovery work on. There it's actually a real advantage that you're somewhat agnostic to the cell source.
Would you consider like a larger market or a market that's less complicated, like?
So great question. And he said, did you consider a market that's maybe less complex and also very large, like food? And there's a company that our scientific founder and co-founder, so Gabor Forgax and Andres Forgax, had founded called Modern Meadow. And I think Andres did a Disruptive Thinkers talk, did he not? Or?
No, I just had dinner with him.
Okay.
Yeah. Hopefully he will sometime.
Yeah, exactly. And what they do is they address the market for food and leather, right? And so you can sort of go onto their website and see how they define the problem they're solving and the solutions they're bringing to it.
Yeah. So rumors are out on the street that you are thinking of like leather. Is that true?
Well, so that's this other company, Modern Meadow, that I described.
Oh,
yeah.
okay.
Yeah. So we focus only on biologic either tissues for transplant or tissues for in vitro use. So living tissues for the biopharmaceutical industry. Modern Meadow is focusing right now on meat and leather.
I'm gonna ask one more question. It's like a math question. See, I can never replicate another Dr. Eric exactly the same like you are. On the outside,
that's probably a good thing.
On the outside, you probably would have one that look the same, right? But when we look at the permutation and combination, they're different, right? So I can inject like a cell sample from you, and then your printer probably would output another Eric. But on the surface level, they look the same. But down to a certain combination of permutation, they are different. So how does that work in say I'm a surgeon, I implant that tissue into a human body during my operation or surgery, right? Would that be still able to assimilate itself with that environment, like with your body, because of a different combination and combination of genes or cell types and stuff like that? You got my question.
Yeah. Well, I mean, so there are really a few different questions in there. One is related to this problem that we discussed earlier of autologous versus allogeneic, and so there are different choices that you can make there, as I described. The other is, look, as you make a tissue, it's never going to be exactly, even if you make it from my cells, it's never, as you say, going to be exactly like my liver. But the beauty of it is it doesn't need to be, right? It still has an awful lot of function. It may not function exactly like my liver, but it functions well enough to replicate, the core things that my liver does. And it is critical that because you bring up, the surgeons who are going to be putting this in. That's an excellent point, right? Someone's going to be implanting these, and therefore you can't have each liver that you make look slightly different and have different, and it's gotta all be, it's got to be a product basically. The surgeon has to know what to expect, not just about how he or she puts it in, but the surgeon needs to know what to expect about the functioning of that organ.
So your error threshold is it like tissue specific? Like if I'm implanting a liver tissue versus a kidney tissue, right? The error, the threshold, this might go wrong.
Right.
Even if I replicate it, but there's like still an error range.
Yeah, there's always going to be some latitude. Even if you get organs for transplant, they have quite different levels of functionality to them. But yeah, within a certain window, you want to know that they're going to operate in that window on a regular basis, and that's what we strive for, right? And it's part of that is making sure you're sourcing the cells from the right places. Make sure you do quality control on those cells as you're growing them up and creating the organs. So they're all solvable problems. But you're right there. When you're dealing with living structures, it's very different from building, as you can imagine, very different from building with bricks and mortar and metal.
Thank you. We hold off questions for now, and if you have more questions, we'll ask them at the end. But if you split off right here and on that side, you go out that way, and we'll do a discussion on civilian or entrepreneurial uses of this technology. And then this group right here, we'll talk about military and government applications. Now, if you really want to do the other one, that's fine too, but I want to make sure all the military guys don't swarm over the military, because I think one of the best things about Disruptive Thinkers is the fact that we tap into both military and civilian. So,
Can we have a military guy here?
yes.
That was cool.
And then Eric's going to go back and forth and answer questions for you, and generally add a dialogue. So, all right. Thank you very much.
Yeah.
Outstanding.
Yeah.